everybody, and welcome to our session on brain imaging genetics in Alzheimer's disease, linking genetics and image-derived endophenotypes. Thank you for coming after the club night on the last day in the morning session. We actually have way more people than we thought would be here, so that's really nice. So this symposium is particularly timely as it focuses on the latest advances in brain imaging genetics and provides a platform to explore some topics such as deep learning, polygenicity of imaging-derived phenotypes, and multivariate assessments of genetics and brain imaging features. So unfortunately, our first speaker had to leave the conference early. So the first one is a recorded presentation, and it is Neda Jahanshad from USC. And she will be giving us kind of a brief overview of brain imaging genetics in Alzheimer's disease. So if you would love to play, perfect. Thanks.
Hello, everyone, and thank you for coming to our symposium on brain imaging genetics in Alzheimer's disease, linking genetics and imaging-derived endophenotypes. I apologize for not being there in person today. I was there at the start of OHBM, but unfortunately had to leave before the end. But thank you so much for sticking it through to the last day, and thank you for coming to our session. My name is Neda Jahanshad, and I'd be giving the introduction to neuroimaging genetics. Afterwards, we'll hear some of the latest and greatest research from three amazing speakers. So we're all very well aware, if you come to OHBM at all, that there is a significant reproducibility crisis involved in neuroscience research and neuroimaging in particular. So we know now that neuroimaging, if you're looking across the brain, you really need large sample sizes and structured analyses and approaches to be able to reproduce findings and have robust results that replicate. Now, it might come as a surprise, but the genetics field has actually been well aware of the reproducibility crisis for many decades. This is because a lot of initial genetic research started by looking at candidate genes. These false positives, if they were false positives, were dangerously costly. And so if you had an underpowered study and you found an inflated effect of a particular gene that you think might be associated with a disorder, you'd be very interested in looking at that mechanism further. And so there was a lot of effort put into potentially making drug targets and preclinical knockouts with these candidate genes that we were looking into that unfortunately did not replicate in different populations. And we were actually underpowered to search the entire genome for genome-wide analyses. And since the early 2000s, when the Human Genome Sequencing Consortium had presented the map of the first human genome, that's when we were really able to look across the entire genome and really step away from these candidate studies that had been done in the past. And what did this Human Genome Sequencing Consortium actually mean for understanding human health? Well, as genetics became more and more popular, we were able to reduce the cost of genotyping dramatically, which led to much larger studies being able to be conducted with the same funds. And these linked and genetic findings really linked to a direct biological mechanism, looking into the specific genes, looking into the proteins, and they can be used hopefully for targeting pharmaceutical treatments for various disorders. For example, in the case of Alzheimer's disease, the genetic architecture is very complex. You'll notice on the x-axis we have here the frequency of particular variations in the genome across the population, where on the left you have very rare mutations, and we go in towards more frequent, common even variations within the human genome. And you'll see that nearly 6% of the population might carry E4 risk allele here. But you'll notice that the very rare mutations, these pathogenic mutations, are highly penetrant, and these are confer much greater risk for disease and even are responsible for early onset Alzheimer's disease, where later onset Alzheimer's is very complex, and there are multiple risk factors in the genome and elsewhere related to the effect. So what do we mean when we say we want to use brain imaging as an endophenotype? Well, by endophenotype, we really mean an intermediate trait. So rather than doing a genetic association analysis with the disease of interest or disease at all, we use quantitative metrics from the brain as this intermediate trait. So endophenotype is defined as something that is associated with a disorder in the population. So for example, in the case of Alzheimer's disease, you might have lower hippocampal volume in patients as compared to control. So that variability in hippocampal volume is related to the disease of interest. And also endophenotype must be heritable, in which case some of the population variance is attributable to genetic factors. And you'll have the endophenotype and the illness co-segregate in families, and families even that are unaffected would have that phenotype or have that endophenotype at a much higher rate than the general population. So what does this actually allow us to do by using brain traits? We can look deeper into the mechanistic processes behind what the genetics are actually doing to give rise to the disease. And maybe we can even uncover genetic associations that affect brain structure and function and might lead to brain traits that might put the brain at risk for other disorders and help drive the search for genes and biological pathways that are related to the disease itself. So what do we mean when we're looking at imaging and genetics? What are we actually doing? So we take, in a very simple association, we take the trait of interest, the endophenotype or the phenotype. In this case, we're using the head size or intracranial volume as the trait of interest, and we're regressing against the minor allele. So as we saw in the Alzheimer's risk diagram, the more towards the right you were going, the more common the genetic variant becomes in the population, and that is the minor allele frequency, how frequent it is. So here we're saying this individual has an AC genotype, and this one has AA. Up here, both individuals have CC genotypes. So we can count the number of minor alleles. In this case, the A is less frequent, so that becomes the minor allele. And we regress that against the intracranial volume that we're looking at. So we run an association. So this is an association between a single genetic variant and a single imaging trait. In addition to looking at a single variant and a single trait, we can take that single variant and map the effects across the entire brain, whether it's across the network, different features, different structural features, different functional features. We can also take a single brain trait and look genome-wide. So not having a particular candidate in mind, but really screening the genome to look at all variants that might contribute to variability in a particular trait. And you can do this, assuming you have the power, across many multiple brain traits, looking at various networks, doing multivariate associations. So all the associations and all the analyses that you're familiar with brain imaging, you can also do with brain imaging genetics for the most part. Now, one big caveat is that these genetic risk factors, especially the common ones, explain very little variability in the population, variability in the population trait. So for example, if we see oftentimes that a single variant might explain 0.1% of the variance in a brain imaging trait, in which case, if you're looking genome-wide, you might need over 30,000 individuals for detecting a robust effect. And to accommodate such necessities in samples and power, especially in relation to particular conditions and disorders, a lot of large-scale multi-site and multi-study consortia formed. For example, some are coordinated, like ADNI, which has the same data collection protocols and study design across many sites, and others are less so and pool data from, for example, the National Alzheimer's Center Consortia pools data from many Alzheimer's disease centers around the US. And we have the United Consortia, Enigma, and Charge that work with data around the world. So before studies like UK Biobank had, individual studies like UK Biobank had over 10,000 samples or had 30,000 samples, you really needed to pool data together from these smaller studies and have these multi-site consortia. In our, so I've been doing a lot of work within the Enigma consortium, so I'll explain a little bit about the studies that we've done in Enigma in relation to imaging genetics. Our first project, published in 2012, was a genetic analysis of the hippocampal volume. So we pulled data from 17 studies, each of which had a modest sample size of between 50 to 1,000 samples, for a total of 8,000 individuals. And we were able to detect an effect of a locus, a genetic locus, on the hippocampal volume, meaning a genome-wide significance threshold after replication. And that genome-wide threshold, if you're assuming you're correcting for 1 million independent variants across the genome, you do a Bonferroni correction of 0.05 divided by 1 million, and it's 5 times 10 to the -8th is the standard single trait genome-wide correction threshold. So we were able to see that while all cohorts involved, and all studies involved in this initial study supported the effect of this variant on hippocampal volume, none of them were significant on their own. So it was only after we were able to pool all the data together and do a meta-analysis that we were able to find significant effects. So we really needed the large sample sizes here. And that study was published in 2012, and since then more and more data sets with imaging genetics have become available, and many more individuals have contributed their analyses to Enigma projects. So we were able to replicate that same study, conduct the study again with more and more samples. So after 2012 and 2015, we did another association of a genome-wide association of the hippocampal volume with now 13,000 individuals, finding a significance threshold of 10 to the -11th. And a few years after that, in 2017, we were able to pool data from the Enigma consortium and from the Charge consortium together for a total sample size of 26,000 individuals, and have a significance threshold of that same locus in the genome that was initially found to be significant in the 2012 paper, continued to be significant and exhibited greater confidence, when we were able to have greater confidence in that finding in 2017 with a p-value of times 10 to the negative 25th. Now, as more and more data become available and we're able to work collaboratively together, we can continue this testing, and we can also look to generalize our findings. So a lot of the a lot of the genetics work, I should say, has historically been in European populations only, because that is where a lot of the data have been collected. Now, as great efforts have been put into collecting data from non-European individuals, we can assess the extent to which the genetic findings in brain traits of European samples might predict the variation or explain the variance in the trait, in that same trait, for individuals of non-European descent. And we can actually see that this is indeed the case across many subcortical volumes. So we are able to get a genetic a summary score from the European individuals and find that it indeed explains nearly 3 to 4% of the variance in several traits in non-European individuals. And as we can do more advanced imaging, we can also apply imaging genetics approaches to the advanced imaging. For example, we recently, within Enigma, pooled data together from about over 10,000 individuals that had at least two scans greater than 6 months apart. We were able to assess the genetics associated with rate of change in the population. And we looked across individuals of developing individuals and also aging individuals, and found that there was, in fact, a locus close to the APOE4 locus that was seemed to have an age-specific effect on hippocampal volume, where the effect got stronger as the age of the cohort got higher. So after about the mean age of the cohort was over around 60, that's when we saw an effect with that locus. And with imaging genetics, you can also look at distinct genetic profiles for different processes in the brain. For example, we found a partially distinct set of genetic loci associated with periventricular versus deep white matter hyperintensity. So there might be different genetics underlying where the white matter hyperintensities occur. And so, in conclusion, we're really working in a greatly expanding field, as both imaging and genetics resources and approaches are advancing. With imaging, we can look deeper into more specific neuroanatomical or functional traits, do a lot of network analyses, multivariate analyses, and really enhance the mechanistic specificity and knowing which brain pathways are affected. And perhaps in the course of disease, you could look at specific brain patterns and subtypes of that disorder and find genetic associations related to those subtype-specific patterns, and really be able to target treatments for those specific subtypes. And also, our understanding of biological pathways and systems are also being enhanced. There's a lot of work being done in functional genetic analyses, where we're looking to map our findings into specific cell types and biological pathways, and do many multi-omics level approaches, which you'll hear about later. And there are these data science methods that are expanding in both imaging and genetics, and they can all be brought together, which again, you'll hear about in the upcoming talks. And all this hopefully will lead to more targeted treatments for disorders In particular, you'll hear a lot about the work being done in the Alzheimer's neuroimaging genetics. So this is really a multidisciplinary and multidimensional field. We hope to have your attention and have you join in on various projects and come join us in the imaging genetics world. So thank you, and hopefully you enjoy the rest of the symposium.